Talking about specific defenses, we know that there are two parts of specific defenses: cell-mediated immunity, which is done by T cells; antibody, or another term for antibody-mediated, is also called humoral immunity, because B cells make antibodies which are released in our body humors, another term for our body fluids. Okay, now we're looking again at the cell-mediated immunity, where we had our antigen-presenting cell, such as a macrophage or a dendritic cell, which comes across the invader, engulfs it, breaks it down, then travels into, usually it finds the T cells that it's looking to see if it has a fit for the antigen. In other words, these have receptors, and these receptors are formed in the development of these T cells after they actually migrate to the thymus. Then they developed a vast variety of receptors that have the ability to recognize a vast variety of shapes that antigens could possibly form. Okay, now, looking here, we actually see, notice this looks very, very similar to what we saw way back here when we were talking about what goes on with the natural killer cell coming across an abnormal cell. Except we said natural killer cells were non-specific. They just look for the absence of certain proteins. And we said cytotoxic T cells do a similar job. So looking back here, here's a cytotoxic T cell that has been activated. Now again, it was activated in this process that formed clonal expansion, and now it actually goes through and looks for infected cells. Usually it's virus-infected cells. So here we see a cytotoxic T cell that has come across a virus-infected cell. Now, this is an actual picture. This is a virus-infected cell here, and here are cytotoxic T cells that have been activated, and they're coming to kill this cell and kill the virus that's inside of it. Now, what it does is, again, it forms the perforin molecules. Notice here are perforin molecules, the process of exocytosis. We release the perforin molecules, we perforate the surface of the cell that's infected. After we perforate the surface, then we actually can release some different chemicals. One of the chemicals is called granzymes. We also have things called lymphotoxins, and what they do, perforating the surface can actually cause it to lyse if we can perforate it enough. But more efficiently, we can actually trigger apoptosis or cell suicide of the cell we release the granzymes in. So anyway, the ultimate goal is for the cytotoxic T cell to find infected cells, because this cell would be displaying the antigen that would be infecting it, and that coming from that pathogen that was infecting the body. Once that pathogen went into this, the cells are all the time taking random samplings of what's going on inside their cytoplasm. So they would not just display the antigen that's infected it, they would also display their own antigens. But along with displaying their own antigens, they're also going to display the antigens of what's infecting it, and that becomes a marker that gives our cytotoxic T cells something to look for, to come across and hopefully kill the cell that is infected. So we see that happening here in this picture. Okay, now really three basic ways. Notice what we have here. We have our infected cell. Our infected cell is displaying, this is a major histocompatibility complex protein. Here's the antigen that it's displaying from the invader. Here's our activated cytotoxic T cell. It has recognized that antigen, and now it's going to release the perforin. It's going to cause the perforations like we see in this picture right here. It's going to release cytokines, cell chemicals, and lymphotoxins, toxins that disrupt metabolism. This helps break down the proteins that would be needed, the enzymes that would be needed for regular cell metabolism. And then the cytokines would actually stimulate the production of enzymes that would actually start eating parts of the cell itself. So here we see three possible ways that we're killing this cell. In this picture, we're seeing two possible ways of killing the cell. But bottom line is that cytotoxic T cell is releasing chemicals that kill the infected cells. All right, now finally we're into looking at our clonal selection as it goes to B cells. So we're actually now looking at what goes on here when we activate our B cells and they start making the antibodies. Because remember, these make cell-to-cell contact. These release antibodies that then attach to things in our body fluids. So notice, though, it really takes T cells called helper T cells that help activate both the cytotoxic T cells and even more directly activate the B cells. So in looking at our description here, notice that I have my invaders here. Notice I have antibodies that serve as receptors. Notice the receptors have different ends because I have to have the correct receptor to basically make a fit with the correct antigen. And so the antibody has receptors, and then it tries to make the fit with the antigen. So the one thing that happens with the B cells is they have to both come across the antigen itself, and then we say it's sensitized, and then after it's sensitized, it has to come across a helper T cell, which releases chemicals that then further activate it into a cloned situation. Notice here's our cloned situation. Once we have those two things, we come across the antigen itself and we get chemicals from helper T cells. Once we've done those two things, we can clone into an army of plasma B cells and memory B cells. Now, the plasma B cells are the cells that actually produce antibodies, and the antibodies then attach to the invaders in body fluids. And then our memory B cells do the same thing basically as our memory T cells. They hang around, so if we have that infection again, then we can actually release more antibodies, but the plasma cells that initially release antibodies, then we can't afford to keep too many white blood cells for every infection that may possibly infect us. So again, there are chemicals released that cause them to go through apoptosis after we've released enough antibodies to take care of the infection. Now, one kind of interesting thing is you can actually give antibodies from one person to another, and they will be effective. So if this person was infected by something and they produce antibodies and they've taken care of the infection and they still have some antibodies left over after they've taken care of the infection, those antibodies will be, they're proteins, they will be proteins found in the plasma of the blood. You can actually take those antibodies from one person and give them to another person, and that will protect them. As a matter of fact, when we make certain anti-venoms, such as anti-snake venom and stuff, what we're actually doing is we're taking the venom from a snake, we're injecting it into, say, a rabbit or something, and its blood is going to form these antibodies. Then we take the antibodies from that, we form our anti-venom. Now we can inject this into somebody, and that basically makes, helps neutralize the snake venom. Okay, we'll talk a little bit more about that process in a few minutes, as a matter of fact. But here we have our memory B cells. One thing, again, we can actually take plasma cells from one person and give it to another person. What we can't do, though, we cannot take the memory T cells and give them to somebody else. Memory T cells, for one thing, they actually are, triggered to attack one type of antigen on our own cells. But in their education process, they learn not to attack our own body cells. And so if we gave these to somebody else, the shape of the receptor may be close to the shape of something on somebody else's cells. So it's kind of like everybody has individual fingerprints, or everybody has individual markers on their cells. So you could possibly give T cells that would have a shape that would attack a normal cell. So we wouldn't give memory T cells to somebody else because it may attack them. But we could give the antibodies from one person to another. Okay. Now, this is a good summary of the way our body takes care of bacteria and the way it takes care of viruses. First of all, viruses are much smaller than bacteria. The what a virus basically does, it likes to basically inject its nucleotides into a cell and trick that cell into making copies of the bacteria. So usually the main thing that viruses do is actually infect cells, okay? And then they reproduce themselves, kill that cell, and spread and do the same thing to other cells until we stop them. Bacteria, on the other hand, tend to multiply out in our body fluids, even though there are some bacteria that can actually go into our cells. Tuberculosis is an example of a bacteria that goes into our cells, but other bacteria just stay outside. So primarily, we're going to kill this with our specific defenses, being if these are inside our cells, is primarily going to be our cell-mediated that's going to kill them, whereas our primarily is going to be our antibodies that's going to kill these. Now, antibodies can also help take care of these, and these can also go into a cell. So if we kill a cell with this in it, then we'll end up killing that also. So both B and T cells can take care of both of them, but primarily T cells kill cells that are infected with viruses, and antibodies attach to these bacteria out in body fluids. Okay, but anyway, getting back to this, let's look at the major difference. Notice bacteria, okay, macrophages are activated. Remember, they're going to be the dendritic cells and the macrophages that come across this and engulf it. They actually, after they engulf it, they will go find the helper T cell, and they will also help activate the cytotoxic T cells. Now, the cytotoxic T cells are the cells that make cell-to-cell contact and actually release the chemicals across into the cells that are infected to kill them. But the helper T cells actually help activate cytotoxic T cells and also activate the B cells. So back here, when we said cytokines from T cells helping activate this B cell here, that's what would be going on from here to here. And then the B cells make plasma cells, and the plasma cells release antibodies. So the antibody production by the plasma cells, then they actually attach to this. And when they attach to it, they actually allow the complement proteins to come attached to it, which help lyse it. They actually mark it so the phagocytic cells can be activated and engulf it much easier. They also clump these things together in two processes called agglutination and precipitation, and they actually neutralize some of these, prevent them from reproducing. Okay, now viruses, on the other hand, oh, by the way, when you take antibiotics, it only works against bacteria. It never works against viruses. So we're not doing ourselves any favor if we take antibiotics for something that doesn't help. Okay, now viruses, they usually infect cells. Now they will move in body fluids between cell and cell, but their main goal is to infect tissue cells so that they can reproduce themselves and spread to other cells. Okay, now viruses can either, once they infect the tissue cells, they actually, way back when we began this, we talked about interferons. So let's just click back here for a second and notice this interferon. And what I told you about that is interferons are released from virus-infected cells, and they protect the cells around it because they trigger the cells around it to produce enzymes that keep the viruses from replicating inside. Okay, so the interferons, basically, it's too late for the cell that was infected, but they spread to the cells around them, and they build up resistance in the cells around them. Now, antigen appearance in cell membranes, remember how when cells are infected by viruses, they tend to display. So here's a cell, and it's displaying the antigen in its membrane here. We can see that. We can even see there's the infected cell, and there it is displaying what's infecting it. Okay, so when we see that appearance in the cell membrane, it will stimulate either the cytotoxic T cells, and that's what we see here. There's a cytotoxic T cell stimulated because this cell's displaying the antigen in its membrane with that MHC complex, so this knows to attack it. Also, remember, it also helps stimulate natural killer cells because the natural killer cells, which were non-specific defenses, if it saw something abnormal going on in the cell, then it's going to kill the cell too. Now, one difference between cytotoxic T cells and natural killer cells, cytotoxic is looking for